As Frank said, my name's Ryan, and I'm here to talk about the crystallographic fragment screening of Enterovirus D68 3C protease. I will give a brief overview of ASAP and XChem in a minute, and then we'll go through how we successfully carried out a fragment screen against the target and how this feeds into the ASAP pipeline. So just a little bit about me. My name is Ryan, and I work in XChem with Frank von Delft. And the focus of us is to conduct crystallographic fragment screens and follow-up screening of compounds within the ASAP pipeline. ASAP itself is a NIH-funded center for pandemic preparedness, focusing on targets such as coronavirus, flavivirus, and picornaviruses. And we're made up of several different projects, of which XChem provides crucial assistance in several of these, primarily target enablement, fragment to lead, and lead optimization. XChem itself is based here at Diamond Light Source, the UK synchrotron, which is in just south of Oxford and just to the west of London. So our target ourselves is enteroviruses. Let's say one of our targets is picornaviruses, and enterovirus forms into this family. Specifically, we're looking at D68 and A71 enteroviruses, and they're typically spread through oral fecal methods, and can lead to several sorts of infections and diseases. And D68 typically causes respiratory illnesses, which can be mild, but can also be severe enough to require hospitalization and intensive care support. And A71 also causes mild illness, but has been shown, but does also cause hand-foot-and-mouth disease, along with some other neuroencephalitis. And just over here, this is a typical hand-foot-and-mouth disease, whereby we have spots that form on the hand, foot, and mouth. A71, in the case of enterovirus outbreaks, although these are not pandemic at the moment, recent outbreaks have been seen quite commonly in Southern and Eastern Asia. And between 2008 and 2014, over 10 million cases were seen in China alone. So it's definitely an area that needs to be explored. So just a little brief background on the viral sort of life cycle. The enterovirus enters the host cell. The viral DNA is released, and this is translated into a large singular polyprotein. The polyprotein itself is then cleaved by three proteases, three viral proteases. These are the 2A, the 3C, and 3CD proteases. So the 2A protease is the first protease which cleaves polyprotein, and this cleaves at this point here. And what this does is it separates out the structural based proteins of the viral polyprotein to the non-structural based proteins. And 2A is also been shown to cleave proteins within the host cell, which are involved in, which enables the polyprotein itself to be enhanced in terms of translated and made, which allows the disease to progress further. And linked to the 2A protease is the 3CD protease. This functions by cleaving up just a little bit further the remaining structural proteins. So 2A causes all the structural proteins to cleave, and 3CD cleaves these up further into the mature proteins. And the target of interest for us is 3C protease. This is of interest because it cleaves a lot of the viral polyprotein. And if we can stop or find an inhibitor of this, then you can quite severely disrupt the viral life cycle. So yeah, it's a good target for in terms of inhibitors. So the 3C protease itself is a cysteine-based protease, and it utilizes a cysteine histidine glutamate triad, as you can see in this image in the bottom. And it's a pivotal part of the viral machinery, enabling multiple of the proteins to be cleaved and become mature. The 3C protease itself tends to be quite highly conserved across different enteroviruses, specifically the catalytic domain, which is to be expected. So we, over on the right-hand side, we have an alignment of different enteroviruses and their 3C sequences. And as you can see, the histidine, the glutamate, and the cysteine are heavily conserved. So what this means is it increases the chance of producing of developing a pan inhibitor. Not to say it will, but it definitely increases the chances that this might be possible. And just to emphasize this a little bit more, what we have here on the left-hand side, sorry, is the D68 3C protease in green and the A71 3C protease in cyan. Apologies for the colors. I realize cyan is probably not the best color for this one, but what we can see is the catalytic site. You have the glutamate, the histidine, and the cysteine. They're all heavily conserved within this region, and they form this on the right-hand side. They form the substrate binding site. So you've got your S1 prime region, S1, S2, S3, and S4, and this is highly conserved across across the 3C enteroviral species. And so, yeah, hopefully maximize chances of producing a pan inhibitor. So on that, there are inhibitors out there, several are known. Probably the more famous one is the Repintravir target. However, there are other targets, GC376, which was a veterinary target, and SG90, which was 98, which is part of another series of inhibitors that were initially developed for future follow-up work. But just going to Repintravir, this was a drug that was developed against the human rhinovirus 3C protease. And as you can see from the structure here, and just referring to the one to the left, you can see how the drug sort of fits into all the different subregions of the catalytic site. So yeah, however, the drug itself was dropped from clinical trials due to efficacy reasons, which was a shame. But what this shows us is that although there's targets out there that aren't necessarily good to go by, it gives us a starting point. It doesn't take anything away from the target being of much interest to us. So what we hope to do with XChem and ASAP is to take our sort of novel starting site and is to use our novel fragment-based screening methods and try and develop a different kind of inhibitor for this region that hopefully has different outcomes. So with this, our fragment-based drug discovery here at XChem, what we do is it's high throughput crystallographic fragment screening, whereby we use the structural data of where fragments bind, such as in this image to the left-hand side. And what we can do is we can build these up into larger molecules that are more potent and bead-like molecules through methods such as on the right here, through sort of fragment growing, linking, and merging, which falls into another part of the ASAP group, into the ASAP pipeline. Our fragments ourselves tend to be smallish, around about 250 Daltons or less. And our libraries sort of span between 500 to 1,000 compound ranges, and we have multiple different libraries which we can utilize. The affinities of these fragments tend to be in the millimolar to micromolar range. However, they're very efficient at entering into the crystal system. And yeah, once we have these, we can use the data to form more follow-up compounds, which we can then use for biophysical methods to sort of back up what we're seeing structurally, and we feed this. It's an iterative process which feeds back into itself. So within XChem, this is just a very brief overline of what we sort of do. So we take crystals, we mount the we soak the crystals with our compounds, our fragments. We harvest the data, and then we refine the structures further to make sure that what we're seeing is there. And then we post our data onto our Fragalysis platform, which is publicly available. So that's the sort of process that we go by. It's a very crude method that I've just explained there, but that's the outline. And XChem ourselves, we have routine users with more than 250 academic projects. And as you can see, as years have gone by, we've increased the number of proposals that we sort of take on. They peaked around 2018, and this is sort of an effect of COVID and whatnot. But data set numbers have increased, and it's a very useful method for fragment-based discovery. So that's sort of a background on XChem and ASAP and the protein. It's the target that we want to focus on. And this is more going into the actual fragment screening itself for the AViDD D68 3C protease. So obviously, before you get any protein, you need to do construct design. This was carried out by Mike Fairhead up at the CMD in Oxford. And he took the viral polyprotein and found the regions where the 3C protease are identified from, and designed a construct based on this. The construct had a non-cleavable C-terminal his tag. It expressed very well, gave good purity. As you can see from here, what this does, it maximizes our chance of getting crystals. And lo and behold, we carried out some initial core screens of on trying to get crystals, and we got a crystal, and it diffracted to about 1.5 angstroms, and the crystal was robust enough. It was a little irregular, but it was a starting point, and one which requires, sorry, here we are, yeah. So this is the condition. We got it down to about, 1.8 angstrom, apologies, 1.8, and crystal reproducibility was about 40%. This becomes more important in the next slide or two, but we had a good starting point, but one which we would hope we could sort of optimize a little further. So the crystal structure itself, there were 2 copies of the protein in the asymmetric unit. So we got chain A in green and chain B in blue. And structurally, very similar, what you would expect. However, what we did observe was that there was a flipping of the cysteine, catalytic cysteine, in chain B. So in green is chain A. So this is how we would expect, by comparing it to other structures in the PDB, this is how we expect the catalytic site to look. You have the cysteine pointing towards the histidine and the glutamate. However, in our structure, what we found was, yes, we had that in chain A, but in chain B, what we actually had was the cysteine had flipped 180 degrees and was pointing away. So this led to a slight change in the catalytic site, which we will come on to a bit later on. But yeah, essentially we have one sort of active form of the protein within the crystal. So as I say, we had initial crystal hits, but we wanted to optimize these further. So in terms of optimizing, we looked at varying the pH, salt, and PEG concentrations, and we also used the addition of crystal seeds from these initial crystal hits. And what we found was actually using these seeds and optimized conditions, we got much nicer, bigger, more robust crystals, which are more friendly for what we need to do. And the The resolution of the crystals was actually a lot better. We've gotten down to about 1.4, 1.5 angstrom. And the reproducibility was up 85%. And what this means is we didn't need to use as much protein and set up as many plates in order to carry out a full screen had we used the previous condition, which gave 40% reproducibility. So this is all about sort of saving amount of protein and time and whatnot. So we have a robust crystal system now. However, we're still not completely there in terms of carrying out a fragment screen. So what we need to do first is identify solvent tolerance. And typically our solvent screens are in DMSO. So we need to carry out a solvent screen to see if our crystals will survive the typical DMSO concentrations that we're going to subject them to. And then following that, if that works, it's then a pre-screen, which is a subset of 100 or so compounds to see if we get any hits, initial hits. And yeah, and then if that comes back positive, we move on to a full screen. So as I say, the first step is solvent testing. So this is where we vary the DMSO percentage and the incubation time that we subject the crystals to. And this typically ranges from 0 to 25% DMSO and between 1 to 3 hours. And what we found from this table up here is that actually the crystals liked DMSO all the way up to 25% concentration, and it didn't actually really change in terms of the incubation time. So, you know, if we wanted to, we would have done 25% soak, 25% DMSO for 3 hours. However, we chose to drop this down a little bit, and this was just to compensate for if sample preparation took a little bit longer. You don't want to lose samples, which might potentially happen. So we just dropped it down, and we decided going forward we would use 20% DMSO for 3 hours, and we should still be able to get a large number of hits based on this. So yeah, we had a robust system that was solvent tolerable, so we carried out a full fragment screen. We did skip the initial pre-screen, and we went for an initial screen, and this composed of 4 of our fragment libraries totaling 1,231 compounds. And what we've got here is just a brief breakdown of how many fragments came from each library. This is not the number of hits, this is the number of fragments that were screened. And the percentage next to them is actually the number of hits that we got from the number of fragments from each screen. And typically we hope to get somewhere between 5 to 10% hit rate. And some of these we were hitting this, one of them we were a little bit lower, and one of them we sort of exceeded this. But yeah, this was good. And in total it gave us 103 hits that we found across 4 main binding sites. Yeah, so we were just shy of the 10%, which we were hoping to get, but you know, very pleased with what we got. So yeah, we got 4. The distribution of the fragments varied over the protein, over chain A and chain B. There were 4 main hit sites that were identified. The catalytic site on monomer A, chain A, which is identified here. This R19, the arginine 19 tyrosine 48 pocket on chain B, which is just up here. The asymmetric unit interface between chain A and chain B, which is number 3. So if you can imagine down here in chain B, this is actually inverted 180 degrees up, and these 2 regions sort of interact as such. And then the last one of interest, well, the last one where a lot of compounds, fragments were found to be was cysteine 60, and this was identified on both chains here and here. So although there were 4 main regions that we found fragments bound, we kind of only focused on 3 of them, and that's the catalytic site, this arginine tyrosine pocket, and C60. So the first one, just looking at, is the catalytic site on monomer A. And what we found was there were 16 fragments that were found to be hits here. The majority of them fell within this S1, S2 subregion. And the catalytic triads, i.e. the glutamate, the histidine, and the cysteine, were somehow involved in binding of, or involved in bond forming with these fragments. So in total, there were 8 fragments that were found in the S1 region and 6 fragments that were found in the S2 region. And there was a couple that were in the S3 region, but we didn't find anything particularly in the S4 or the S1 prime region. But this definitely gives us a starting point to build from, and yeah, we feed this into the fragment to lead, fragment optimization, lead optimization in the next step, in the next project, P3. Yeah, the other main hit region was this arginine 19 tyrosine pocket on chain B. So what we found here was actually the majority of the fragments were either aromatic or contained ring features. And what we found was actually there was multiple pi cation interactions that seemed to be occurring in this region and sort of stabilizing why these fragments were found here. Not entirely sure if this pocket is of biological relevance. The main reason for that is, if you remember on the previous slide where I said that chain B, the catalytic site, the catalytic cysteine, had flipped 180 degrees. This may have caused the conformational chain. The catalytic site is somewhere just over here, I believe. On the left-hand side, it's somewhere over here. So I don't know if it's caused some form of conformational shift, which may be may have sort of enabled all these compounds to end up being here. The only reason I say this is because we don't see it on chain on monomer A. Sorry, something's popped up on my computer and I now can't do anything. Okay. And then finally, the third region that we sort of found to be of interest was this surface pocket region on both monomers of the cysteine 60. So you can see here on the right hand side, here's the cysteine. It's in 2 different conformations, the cysteine, depending on which fragment was found where. But most of the fragments themselves interact somehow with either this cysteine or some of the surrounding peptide backbone region. But this could be an allosteric site potentially, or it could just be a place where fragments are binding. But it's an area which, again, we will try to exploit at a later date in the following projects. So yeah, as I say previously, all this data is available. It's all put on the it's all put onto our web server called Fragalysis, and it's publicly available for people to go out and look at and see where our hits are. Just to break down, you have the different sort of target region, hit region sites, and then over here, just a bit below, you can actually see individually where certain fragments are located, and any sort of bonds or interactions that are occurring. It's very useful, and it's what we use it's what we feed into the pipeline, and is what the other project cores also use in terms of developing fragments further. So yeah, as a next step, fragment progression is going to be carried out using machine learning, computational chemistry, using various strategies from Project 3, the fragment to lead and target validation, lead to optimization, and Project 4, sorry. Lead to optimization itself is an iterative process whereby these new compounds that are made, we will be looking at, they will be fed back into the structural biology core, and we'll be looking at them through the structural data. However, in addition to this, we also have the use of biochemical and biophysical assays. Biochemical assays have been developed and will be performed to validate these follow-up compounds. And yeah, the data with that and the structural data will be sort of fed back in, and we can use that to see where we need to build and possibly go further. So in terms of the assays, the biochemical assay that's been developed, this was developed and it's been conducted and tested by Charlene Giroux up at the CMD in Oxford, and utilizes fret shift signals. So what we have here is the natural peptide substrate, which is dual labeled, a bit more obvious over here. So we have this EDANS donor fluorescent signal, which is currently quenched. However, when we subject this peptide, the natural peptide sequence, to the 3C protease, what we should get is cleavage between the glutamate and the glycine. And what you should get then is a fret shift, a signal shift. And what we can do is we can use this to, we can see if the signal changes, we know that the peptide has been cleaved. Whereas if it doesn't, then we know that the peptide, the protease has been inhibited in some form or description. So what we can use is this assay has been tested with controls, and we've seen this whereby we don't get a shift when the inhibitors, when the control inhibitors are used at higher concentration, and then as you go down in concentration, we see that there is a shift in the signal. And what we can do is we can utilize this in with any follow-up compounds and hit to lead compounds. So I say it will feed in with the structural data, which will be useful for the chemists. So all of this data that we have, what do we do with it? It's all available on the ASAP webpage. We regularly update this. So on the right-hand side is just a pipeline overview. This is on the website. You can use this QR code here to see this. And this just gives an update on where we are with certain. These are all of our targets that we are currently taking part in, and we update this regularly to say where we are with them. And D68 is up to sort of the fragment screening point and the lead optimization point now. Sorry, hit to lead next. But also we have these target enabled packages, which gives all the information in terms of methods and what was used, the results. And this is available for anyone to access. And what we have here at the top of each target page is just a nice sort of brief outline on where we are. So green is complete, orange is brown, whatever color that is to the point of it's underway or it's near completion, and then white is to be done. Oh, that's the end. And yeah, so just a big thanks to everyone who's part of ASAP. There's a lot of us. There's a lot of different groups. Big thanks to Diamond and the CMD lot. Yeah, we all pull in a shift and we all manage to get these fragment screens out. We help each other. They're not trivial things to do. They are quite labor intensive, and especially data processing. But yeah, thanks for listening.